Welcome you, madam. Shall we
Well,
proceed, madam?
okay, sir. Well,
So bacteriology, systemic bacteriology. No?
yes, sir. Systemic
Yeah,
bacteriology
part
part.
two. Part two. Thank you, madam. Please proceed.
All right, sir. Hello, everyone. Welcome to Tawhid Foundation at Allied Healthcare Sciences, Bangalore. Today's class will continue with systemic bacteriology part 2. In last class, we studied about gram-positive and negative cocci and gram-positive and negative rods. Today, we'll begin with other bacteria. We'll start with Klebsiella. So this slide introduces the genus that is Klebsiella, which are gram-negative, non-motile and shaped bacteria with a thick protective polysaccharide capsules. These bacteria are commonly associated with serious human infections, particularly in immunocompromised or hospitalized individuals. They are a significant cause of hospital-acquired infections such as Klebsiella pneumonia, urinary tract infection, septicemia, and wound infections. So its cultural characteristic is they can ferment glucose, lactose with the production of acid and gas. Disease caused by Klebsiella pneumonia. Klebsiella can cause a spectrum of diseases depending on the site and patient susceptibility. The most notable is pneumonia, often described historically as Carangelli sputum due to the bloody mucoid nature of material. Apart from pneumonia, Klebsiella can cause severe urinary tract infections, wound or surgical site infections, and in some rare cases, it can cause meningitis. So, these infections may present at greater risk in healthcare settings due to antibiotic resistance. Second is Proteus. Proteus genus, another group of gram-negative bacilli from the Enterobacteriaceae family. Proteus species, especially Proteus mirabilis and Proteus vulgaris. These two are the famous for their extreme motility, that is swarming on agar, production of urease and notably fishy odor in cultures. Infections are common in both community and healthcare environments, especially involving the urinary tract. Again, its cultural characteristics. So here the focus is on laboratory identification. Proteus demonstrate a distinctive this swarming growth pattern on non-selective agar media caused by concentric circles of bacterial spread. The ability to decompose urea into ammonia is key for laboratory differentiation. Proteus also produce hydrogen sulfide gas, that is H2S, so visible as blackening in specific media like triple sugar ion TSI agar. Proteus mirabilis. So Proteus is commonly implicated in urinary tract infection, notably catheter-associated cases. In hospitals, it can also cause wound infections, particularly in diabetic or immunocompromised individuals. A special complication is the formation of kidney stones due to alkalization of urine from urease activity, predisposing patients to chronic or recurrent infections. So UTI is the main cause of protease, and bacterial urease to produce ammonium salt is the basic thing. This result in alkaline urination pH and increased risk of urinary tract infections. It can also cause septicemia. Treatment, you can tell protease are highly resistant to antibiotics. Sensitivity test should be done before treatment, and Protease mirabilis is inhibited by the most common antibiotic, that is penicillin. Other members of group included aminoglycoside and cephalosporins. Next is Salmonella. So the genus Salmonella, a group of gram-negative motile rods known for causing a variety of GIT, that is gastrointestinal and systemic illness. Salmonella can be categorized into typhoidal and non-typhoidal strains, with the former Salmonella typhi and paratyphi. These two, Salmonella typhi and paratyphi, responsible for enteric, that is typhoid fever in men or women as well, and the latter cause self-limited foodborne gastroenteritis. It can also cause food poisoning. Other species of Salmonella Salmonella, which cause enteric fever. So enteric fever is a severe systemic illness with a long incubation period and stepwise fever. After ingestion and gut invasion, the bacteria enter the bloodstream, causing bacteremia, splenomegaly, and multi-organ involvement. So it is a life-threatening condition that required rapid medical attention, most common in areas with poor sanitation or contaminated water. So they cause a variety of condition which is referred as salmonellosis. First is enteric fever, second is enterococcylitis, third is bacteremia and septicemia. So we'll start with typhoid fever, that is enteric fever. It is generally produced by Salmonella typhi and Salmonella paratyphi. Types is A, B and C. Sources habitat of infection is generally stool or urine cases. Infecting dose is about 10 to the power of 5 organisms. So the organisms enter the body via the oral route in contaminated food or drinks. Examples include raw vegetables, fruits, raw shell, fish, and other dairy products. After injection, the organisms multiplies in pairs patches, and lastly, it passes through the lymphatics to the bloodstream, which cause bacteremia that persists for last which will last for about 1 week. It then disseminates to the kidney and is excreted in urine and to the liver in bile to reach the intestine. So enteric fever is a severe systemic illness with a long incubation period. Beyond enteric fever, salmonella can also cause focal infection and asymptomatic carriage, especially in gallbladder carrier side. These bacteria are adept at surviving in both animal as well as human hosts, making control and prevention challenging without strict public health measures. So the spectrum of disease can range from mild gastroenteritis to severe invasive disease in vulnerable conditions. Clinical features. So enteric fever caused by Salmonella typhi and paratyphi typically present with a step ladder pattern of rising fever, headache, malaise, abdominal discomfort, and sometimes a characteristic rash, which will called as rose spots on the abdomen. Other symptoms include hepatosplenomegaly, constipation in early stages, and later diarrhea. So the incubation period will last up to 10 to 14 days. Fever, all these conditions will occur in if the person is infected by salmonella. After recovery, some individuals continue to harbor salmonella in their tissue as chronic carriers. They can carry the organism in gallbladder or urinary tract, which excreted later in the stool or passed in urine. Such individuals should not work as food handlers so that they'll not infect others individuals. Diagnosis. So, diagnosis primarily begins with blood cultures during the first week of illness. Since bacteria are most likely to be found in bloodstream at this stage, the Vidal test is a serological test detecting antibodies against ONH antigens of Salmonella but is less reliable by itself in later stages. Stool and urine cultures become useful for isolation of this bacteria. So, diagnosis of enteric fever is through isolation of organism from all these samples, either blood, urine or stool sample, and detection of antibodies in serum of the patient. Serological tests include Vidal test, as I mentioned, O and H antigens of salmonella will be used. Prevention, you can use sanitary measures to avoid contamination of food and water, and these carriers should not handle workplace while cooking food. Treatment, the drug of choice are chloramphenicol, ampicillin, and trimethoprim sulfamethoxyl. Salmonella enterococcylitis. So symptoms of Salmonella enterococcylitis usually include abrupt onset of fever, abdominal cramps, nausea, vomiting, and diarrhea, which sometimes be bloody. So symptoms typically last around 2 to 7 days, and most cases are self-limiting, though dehydration can occur, especially in young children and elderly individuals. So many members of Salmonella are responsible for this condition. The most important one are Salmonella typhimurium and Salmonella enteriditis. These organisms are the common pathogens which will include in animals or as well as birds. Transmission organism is transmitted from improperly cooked meat of infected animals, for example, chicken or eggs of infected birds, or from food contaminated with rat or excreta. Symptoms Symptoms of Salmonella enterococcitis usually include abrupt onset of fever, abdominal cramps, nausea, vomiting, and diarrhea, which will include blood. The disease is characterized by these symptoms. The condition occurs after multiplication of organisms in intestine. That is, for example, it is an infection and not due to toxin. So the incubation period will last up to 12 to 18 hours, which will allow multiplication of infection. Recovery follows within 1 week of period. Salmonella septicemia. So, in certain high risk population, especially immunocompromised or those with underlying diseases, health condition, Salmonella can cause primary septicemia. This involve bacteremia without major intestinal involvement, which may lead to localized infection elsewhere, enterocarditis, osteomyelitis, and meningitis. So these osteomyelitis, pneumonia, meningitis, or endocarditis can be early invasion of the bloodstream with possible focal lesions. Shigella. So this slide introduces Shigella, a genus of gram-negative non-motile rods closely related to Escherichia coli, that is E. coli. Shigella species are highly infectious, needing only a small inoculum to cause diseases, and are responsible for bacillary dysentery, that is shigellosis, and acute bloody diarrhea. So natural habitat will limit to the intestinal tracts of humans and other primates where they cause bacillary dysentery. They are closely related to E. coli, and they produce non-lactose fermenting colonies on McConkey agar. Antigenic structure. So according to somatic O antigens, Shigella are divided into 4 serogroups or species and numerous serotypes. So Shigella is classified according to these 4 main groups A to D. Corresponding to Shigella is Shigella dysentery, Shigella boydii, flexneri, and soni. So in this there are 13 serotypes, and second one it include 18 serotype, third one include 6 serotypes, and last one include 1 serotype. Pathogenesis of shigellosis centers on the ability of Shigella to invade and destroy colonic epithelial cells by producing Shiga toxin, which inhibits protein synthesis, which will lead to mucosal ulceration, inflammation, bleeding, and the classical dysentery syndromes. So these are the pathogenesis caused by Shigella, and the infection occurs through the ingestion of contaminated food or drink, and the infection is limited to the gastrointestinal tract without blood invasion. They invade through mucosal epithelium, which will cause microabscesses in the wall of large intestine, and terminal ileum, which will lead to necrosis of mucous membrane, superficial ulceration, bleeding, and formation of pseudomembrane on the ulcerated area which is infected. This consists of fibrin, leukocyte, cell debris, necrotic mucous membrane, as well as bacteria. Pathogenesis of Shigella centers on the ability of Shigella to evade and destroy colonic epithelial cell by producing this toxin. The Ministry of Diagnosis is stool culture, particularly from fresh diarrheal specimens. Specific media can help isolate Shigella, which typically does not ferment, that is lactose, and is not motile. So recovery occurs spontaneously. Shigella produces 2 types of toxin. First is endotoxin and second is Shigella dysentery exotoxin. So Shigella cause diarrhea, usually bloody stools, fever, stomach aches. These are the symptoms which will cause in Shigella. Specimen include fresh stool samples, mucus flakes, and rectal swabs. Large number of fecal leukocytes and sub-red blood cells are seen microscopically in specimens. The material are then streaked on media, that is McConkey agar. Non-lactose pale color colonies will be identified according to their morphology. Biochemical test and serological test agglutination with specific antisera will be done to find out the Shigella in specimens. Next is Pseudomonas. So this slide introduces Pseudomonas, particularly Pseudomonas aeruginosa, a gram-negative aerobic motile rod with polar flagella often associated with opportunistic infection in hospitals. So Pseudomonas aeruginosa produces distinctive pigments, that is pyocyanin and pyoverdine. So and is not notable for its resistance to multiple antibiotics and disinfectants, making infection control a significant challenge. So nearly 30% of hospitalized patients show this colonization of Pseudomonas aeruginosa is the major human pathogen of the third most common cause of nosocomial infection after Staphylococcus aureus and Escherichia coli. Antigenic structure. Pseudomonas, particularly Pseudomonas aeruginosa, display both O, that is somatic, and H, that is flagellar antigens, contributing to its identifiable serotypes. These antigens are important in immune evasion and pathogenesis as well as epidemiological typing in outbreak investigations. So pili, it will extend from cell surface and promote in motility, that is attachment to host epithelial cell. PS capsule is responsible for the mucoid colonies which is seen in culture from PTS with cystic fibrosis. LPS exists in multiple immunotypes, which is responsible for many of the endotoxic properties of the organisms present. Extracellular enzymes, most Pseudomonas aeruginosa produce two types of enzyme, that is exotoxin and endotoxin. So exotoxin A, which causes tissue necrosis and it is very lethal for animals. It blocks protein synthesis like diphtheria toxin. Other examples include elastasis, proteases, and second type of hemolysins. Pathogenicity, an important cause of hospital-acquired infection. Pseudomonas aeruginosa is recognized for its remarkable ability to cause disease in weakened hosts. It produces various virulence factors including exotoxin, elastases, and enzymes that degrade tissues and impair immune responses. These organisms is a common cause of pneumonia in cystic fibrosis patients, burn wound infections, UTI infections, and sepsis in immunocompromised individuals due to its resistance and ability to thrive in most hospital environments. These are the infections: chronic infection associating with dwelling catheters, patients in hospitals, wound and burn infections, septicemia, all these eye infections secondary to trauma or surgery. These are the pathogenicity. Diagnosis of Pseudomonas is identified in the lab by its characteristic growth. Colonies may have a blue or green color from pyocyanin pigment and a distinctive grape-like odor. It can be isolated from clinical samples using selective media and further distinguished by its biochemical properties such as oxidase positivity and non-fermentive metabolism. It does not ferment lactose and it is oxidase test positive. Genus Vibrio. So Vibrio includes curved gram-negative rods commonly found in aquatic environments. Vibrio cholera, the most medically relevant species, is the agent of cholera and acute diarrheal illness. Other members like Vibrio parahemolyticus and Vibrio vulnificus can cause seafood-associated gastroenteritis and wound infections respectively. So vibrios are widely spread in nature, mainly in water, contaminated water, and habitat. It can also included in feces or carriers. It is gram-negative, comma-shaped bacilli arranged in pairs or short chains, giving a spiral appearance in microscopic images. Toxin produced by Vibrio. Vibrio cholera produces cholera toxin, that is CT, a potent enterotoxin that activates annihilates silase, resulting in increased secretion of water and electrolytes in intestinal epithelium. This mechanism is responsible for the profuse rice water diarrhea characteristics of cholera. Other toxins and hemolysins also play a role in the virulence factor of cholera. Vibrio. Pathogenesis. So after ingestion of contaminated food or water, Vibrio cholera attaches to the small intestine's mucosa and it bypasses stomach acidity, which will multiply without invading tissue. So cholera toxin is secreted, causing massive watery diarrhea and rapid dehydration, which can be fatal if it is kept untreated. IP, that is incubation period, is short, less than 1 to 4 days. Infection is restricted to intestine with no blood invasions. Organisms attached to the microvilli of border of epithelial cell where they'll do multiplication and liberate cholera enterotoxin, which will bind to gangliocyte receptors of the mucosal cells. So after a lag period, which is of 15 to 45 minutes, adenyl silase is activated and the CAMP concentration inside the intestinal cells are increased. So after increased intracellular CMP result in excretion of electrolytes such as chloride and bicarbonate ions along with massive quantities of water will be passed. So severe diarrhea and vomiting may lead to dehydration and death. So convalescent carriers occur, but chronic carriers are very rare. Diagnosis of cholera. Diagnosis is confirmed by isolating Vibrio cholera from stool samples using selective media like thiosulfate citrate bile salt sucrose, that is TCBS agar. This TCBS agar. Microscopy is less useful, but rapid diagnostic tests and serological identification assist in outbreak settings. Proper identification is critical for epidemic management and patient care. So stools are inoculated on alkaline peptone water and subcultures are made from the surface pellicle after 6 to 4 hours of incubation on TCBS agar. Colonies were identified through morphology, a wet mount stain which is examined for motility. Smear stain shows gram-negative, comma-shaped bacilli in shape. Biochemical activities including cholera, red reaction, agglutination with specific anti-cholera sera, that is polyvalent, anti-inaba and anti-ogawa sera. These include as biotic treatment include intravenous fluids to correct the fluid and electrolyte balance. Antibiotics have a secondary role in treatment, that is tetracyclines are the most effective. So the cornerstone of cholera treatment is prompt rehydration therapy, either orally or intravenous, that is IV, to counteract fluid loss. Antibiotics, all these antibiotics can shorten illness duration, but are secondary to rehydration in priority. So severe cases may need supportive care to manage shock or metabolic disturbances. Prevention. So no specific prophylaxis is available for cholera. Proper hygiene measures should be followed to avoid infection. Treatment, restoration of fluid and electrolyte balance in severe cases. Antibiotics, all these antibiotics will show effective treatment against cholera. Next is spirochetes. This slide introduces the broad group of spirochetes, which are long, thin, spiral bacteria with unique motility due to internal flagella, that is axial filaments. Important genera include Trypanoma, Leptospira, and Borrelia. So these organisms cause a variety of systemic diseases such as syphilis, leptospirosis, and Lyme diseases, often with chronic or multi-stages. So its morphology, spirochetes have unique helical structure, central protoplasmic cylinder bounded by the cytoplasmic membrane, cell wall of similar structure to that of gram-negative bacteria, the axial filaments regarded as internal flagella. Its habitat, they are free living and non-pathogenic in nature, but few will cause important human diseases. It is consists of three genera, as I mentioned: Trypanoma, Borrelia, and Leptospira. Trypanoma will cause syphilis, yawns, and pinta. Borrelia, it will cause a relapsing fever and Lyme diseases. Leptospira cause leptospirosis, that is Weil's diseases. The genus Trypanoma includes few pathogen members, that is Trypanoma pallidum is the most important and is the causative agent of syphilis. So syphilis disease is caused, this causative agent is Trypanoma pallidum. Its morphology, it is cylinder in shape with spiral coils regularly spaced 1 μm apart. They are actively motile by means of an eggshell filament. So, the causative agent of syphilis is Trypanema pallidum. The triponomies replicate by transfer fission. The cells remain joined to each other. These species are not stained with gram stain but can be seen by dark ground microscopy. In unstained preparation, fluorescent label antibodies or silver stain is needed to demonstrate the spiral morphology of Trypanoma pallidum. Disease syphilis. So syphilis progresses through stages, that is primary at the site of infection, secondary systemic rash and mucosal lesions, latent, that is asymptomatic but persistent infections, and last is tertiary, that is gummatous lesions, cardiovascular and neurological complications. So congenital syphilis occurs via placental transmission causing severe fetal outcomes. Primary, secondary, and tertiary. We'll go one by one. Primary syphilis, bacteria enter the body through tiny breaks in skin or mucous membranes. The Trypanoma pallidum enters the lymphatics, regional lymph nodes, and involved and cause bacteremia. Cancri develops at the site of inoculation, which is around 6 to 12 weeks after exposure to infection, as a papule on genitalia, which will cause ulcerates. It is not painful, but it will heal spontaneously. Secondary syphilis, lesions occur after 6 to 12 weeks after the appearance of cancri and are characterized by generalized manifestation, for example, skin rashes. Palladium is found in large number in lesions in both primary as well as secondary stages and are highly infectious. About 25% of patients are cured easily, but 25% progress of the tertiary stage is quite tricky. Tertiary syphilis, cellular immune responses to Trypanoma palladium and its metabolic products. It is characterized by its appearance of stomata in internal organs around 5 to 40 years after initial infection if it is not treated properly. CNS, central nervous system involvement, as manifested by tabes dorsalis and paralysis. Cardiovascular lesions causing aortic aneurysm, which are very common in syphilis. Next is Mycobacteria. Mycobacteria are acid-fast bacilli with unique cell walls containing mycolic acids. This genus include important pathogens like Mycobacterium tuberculosis and Mycobacterium leprae, notable for causing chronic granulomatous infections. So mycobacteria, it is rod in shape aerobic bacteria that does not form spores. Several members of this species cause diseases in men and animals, while others are saprophytes. The commoner species which are classified are typically tubercle bacilli, that is Mycobacterium tuberculosis and Mycobacterium bovis. The atypical or environmental Mycobacteria, which are opportunistic pathogen, that means that will infect under their favorable conditions. It is generally found in water, soil, animals, and men without evidences of disease. Next is Mycobacterium leprae. Morphology, they are thin, straight, or slightly curved rods. It is the causative agent of tuberculosis. It will primarily infect the lungs but can involve any organ. The bacteria are slow growing, requiring specialized media and lengthy incubation for culture. Transmission occurs mainly via aerosol droplets from infected individuals, so they cannot be stained by simple stain due to their high lipid content, which will include mycolic acid, that is long chain fatty acid, waxes, and phosphatides. In the cell, these lipids are largely bound to proteins and polysaccharides as well. When it is stained by Ziehl-Neelsen stain, they appear as thin pink rod which is arranged singly or in small group of pairs. So once it is stained, they resist decolorization with 20% of H2SO4, that is sulfuric acid and alcohol. Cultural characteristics. So this slide covers the cultural characteristics of Mycobacterium tuberculosis. It grows on Livingston ginseng, that is LJ medium, as slow growing, rough, buff-colored colonies. Its acid-fast property is demonstrated by Ziehl-Neelsen staining. Cultures will take around 4 to 8 weeks to become visible due to their slow replication process. Pathogenicity of Mycobacterium tuberculosis. The pathogenicity involves inhalation of bacilli that reach alveoli, triggering macrophages ingestion, but resistance to destruction, which will lead to granuloma or tubercle formation. So reactivation or progression cause lung tissue damage and systemic spread of infection manifesting as primary and secondary TB. That is tuberculosis. Both Mycobacterium tuberculosis and bovis are pathogenic to guinea pigs. When injected to a local nodule develop ulcerates within 4 to 6 weeks period of time. It is associated with enlargement of draining nodes. Rabbits are most susceptible to Mycobacterium bovis, which causes generalized fatal infection, than tuberculosis, which causes mild local lesions. This disease can infect any organ of the body, but it especially uh, occurs uh, when infected with Mycobacterium bovis, and it is transmitted by ingestion of milk from infected cattle and cause intestinal tuberculosis. Tuberculosis produces no recognized toxin. This disease results from the multiplication and local invasion at the site of infection. These organisms is capable of multiplying intracellularly and hypersensitivity, which will lead to tissue damage. And the pathogenicity also involves inhalation of bacilli that will reach alveoli, and it causes infection. Infection with Mycobacterium tuberculosis confers on the second condition, that is resistance and hypersensitivity. Immunity and hypersensitivity are the features of CMI2 tuberculosis. Transmission occurs through inhalation of airborne droplets containing bacilli from active TB patients. When close contact and overcrowded environments increase the risk of infection. Immunosuppressed individuals are especially vulnerable to infection and disease progression. So human TB is caused by Mycobacterium tuberculosis and Mycobacterium bovis. Diagnosis will depend on detection and identification of TB in their isolation from specimen which are pathogenic. Specimen TB can affect every tissue in the body. So type of specimen for examination varies wildly. Sputum is generally given for identification of TB. For PT diagnosis, urine sample for renal tuberculosis, CSF for meningeal tuberculosis, stool for intestinal tuberculosis. Like this, specimens will vary according to the site of infection. The following steps are made for detection or isolation of TB from sputum. So again, it will the specimens will be stained with Ziehl-Neelsen stain. The detection of AFP in sputum will given fairly strong indication of PTB, but are detected only if they are present in large numbers. So the smear stained by oromine O and examined by fluorescent microscope for yellow fluorescent tubercle bacilli. Negative specimens are repeated for 3 successive days and re-examined after concentration. So there should not be any confusion whether the patient is infected with TB or not. Treatment requires a prolonged multi-drug regime, usually at least 6 months, combining isoniazid, rifampicin, pyrazinamide, and ethambutol to prevent resistance and ensure bacterial eradication. Adherence to treatment and monitoring are essential for cure. So this is because most bacilli are found intracellular. Case material interfere with these drugs, and in chronic lesions, TB bacilli are not dividing. That is, they are metabolically inactive, hence resistant to drugs. Combination of drugs due to prolonged course of treatment needed, which will lead to toxicity. Rapid emergence of resistant strain. First is combination of drugs should be used to reduce toxicity and its resistance. The drug of choice are isoniazid, rifampicin, streptomycin, para-aminosalicylic acid, and ethambutol. These which are mentioned earlier. Prevention, public health measure, maintain hygiene, early DX of cases and their treatment until they become non-infectious, control of infection milk from milk by pasteurization of milk. Second is vaccination. Live attenuated vaccination primarily involves the Bacillus calmette-guerin (BCG) vaccine. BCG is a live attenuated strain derived from Mycobacterium bovis and is mainly used to protect infants and young children from severe forms of TB such as mild TB and TB meningitis, while not fully protective against pulmonary TB in adults. So it remains a critical component of TB control program, which is worldwide. Next is Mycobacterium leprae. So Mycobacterium leprae, the causative agent of leprosy. Hansen disease, it is also known as Hansen's disease. This acid-fast bacilli has not been cultured in artificial media and requires long incubation period in specialized hosts, example armadillosis, or experimentally in food pads of mice. So for propagation, it primarily affects skins and peripheral nerves, which will lead to characteristic lesions and disabilities. So its morphology, they are similar in size and shape to tuberculosis bacilli, and they are acid fast. Only 5% H2SO4 is used for decolorization. They are also alcohol fast. Leprosy, Hansen's disease caused by Mycobacterium leprae. Pathogens replicate within skin, endothelial cell, and the Schwann cells of nerves. Two distinct forms of leprosy is tuberculoid and lepromatous, which will exist. Tuberculoid leprosy. So it represents the milder form with well-formed granulomas and strong cell-mediated immunity. Lesions are very few, well demarcated, often hypopigmented, and associated with peripheral nerve enlargement. Disease progression is slow and nerve involvement is localized. So, leprosy is the more severe multibacillary form, which is characterized by widespread lesions, nodules, and plagues with heavy bacterial loads. There is a weak cell-mediated immune response, which will lead to progressively extensive nerve damage, which will form deformities and disability in individuals. Treatment. Dapsone with or without rifampin is used to treat the tuberculoid form of disease. Clofazimine is added for the treatment of lepromatous form. Therapy is given for long period of time. Prevention and control include chemoprophylaxis with dapsone for exposed children is generally recommended. Disease is controlled through the prompt recognition and treatment of infected individuals. So summary, in today's class, we learned about Klebsiella, Proteus, Salmonella, Shigella, Pseudomonas, Vibrio cholera, Trypanosoma pallidum, Mycobacterium tuberculosis, and Mycobacterium leprae. Klebsiella will cause pneumonia and UTI septicemia in individuals. Proteus, there are two types of Proteus, Proteus mirabilis. So UTIs, it will also form kidney stone formation in individuals. Salmonella, it will cause typhoid fevers. There are three types of salmonella which will cause diseases: enteric fever, enterococcitis, septicemia. Shigella, it will cause bacillary dysentery. Pseudomonas, it is an opportunistic infection when individual is infected with burns, wound infection, sciatic fibrosis, generally in hospitals. Vibrio cholera, cholera with rice watery stools. Trypanosoma pallidum, it will cause syphilis. There are three stages of syphilis: primary, secondary, and tertiary. Mycobacterium tuberculosis, pulmonary and extrapulmonarily TB. Mycobacterium leprae, it will include tuberculoid and Leprometrius leprosy. Question for 3-mark question, there is 10 questions. We'll start one by one. First, name 2 important diseases caused by Klebsiella pneumonia. Second, mention 2 cultural characteristics of Proteus species. Third, define row spots and enteric fever. Fourth, list 2 clinical features of Salmonella enterococci. Fifth, what is the infective dose of Shigella and why is it clinically important? Sixth, mention 2 pigments produced by Pseudomonas aeruginosa. Seventh, write any 2 toxins of Vibrio cholera and their functions involved. Eighth, name the stages of syphilis which will caused by Trypanosoma pallidum. Ninth, list 2 specimens collected for the diagnosis of tuberculosis, like sputum or any samples of the infected area you can mention here. 10th, mention 2 differences between tuberculoid and Leprometrius leprosy. For 5-mark questions, first, explain the pathogenicity and disease by Klebsiella pneumonia. Second, write an account of cultural characteristics and diseases of Proteus. Third, describe the pathogenesis and clinical features of enteric fever caused by Salmonella typhi. Fourth, discuss the diagnosis of enteric fever caused by enteric fever, which will include blood culture, Vidal test, etc. In detail, you have to mention. Fifth, explain the pathogenesis of Shigella dysentery with reference to Shigella toxin. Sixth, describe the pathogenicity and clinical infections caused by Pseudomonas aeruginosa. Seventh, write a note on the pathogenesis, clinical features, and diagnosis of cholera. Eighth, discuss the stages of syphilis caused by Trypanosoma pallidum. Ninth, write an account of the pathogenesis, clinical features, and treatment of tuberculosis. Last is differentiate between tuberculoid and Leprometrius leprosy with reference to its pathogenicity, clinical features, and treatment. So that's all for today. With this class, we completed the syllabus for BSc MLT first year students.
Yeah, you nicely completed, madam. Systemic bacteriology for
Yes,
BSM. Thank you so much, madam. I think today we have completed first year BSc MLT syllabus.
Syllabus is, yes, sir, completed. So from next
Excellent.
class, we'll start with second year students, sir.
Yeah. Thank you, madam. Thank you so much.
Thank you, sir.
So tomorrow we'll start here day after tomorrow.
One day I'll go through with the syllabus, sir, and
Yeah.
check for the notes and everything. And from day after tomorrow, we'll start, sir.
Done.
Inshallah.
No issue.
All right,
Thank
sir.
you.
Thank you, sir. Thank you.